Biotechnology and its application. Biotechnology, as you would have learned from the previous chapter, essentially deal with the industrial scale production of biopharmaceutical and biological using genetically modified microbes, fungi, plant and animals. The application of the biotechnology includes therapeutics, diagnostic, genetically modified crops for agriculture, processed food, bio remediation, waste treatment and energy production. Three critical research areas of biotechnologies are: 1. Providing the best catalyst in the form of improved organism, usually a microbes or a pure enzyme. 2. Creating optimal conditions through energy engineering for a catalyst to act. And 3. Downstream processing technologies to purify the protein or organic compound. Let us now learn how human beings have used biotechnology to improve the quality of human life, especially in the field of food production and health. Biotechnological application in agriculture. Let us take a look at the three options that can be thought for increasing food production. 1. Agrochemical based agriculture. 2. Organic agriculture. And 3. Genetically engineered crop based agriculture. The Green Revolution succeeded in tripling the food supply, but yet it was not enough to feed the growing human population. Increased yield have partly been due to the use of improved crop varieties, but mainly due to the use of better management practices and use of agrochemicals, fertilizer and pesticides. However, the farmer in the developing world, agrochemicals are often too expensive and further increases the yield with existing varieties are not possible using conventional breeding. Is there any alternative path that our understanding of genetics can show so that farmer may obtain maximum yield from their fields? Is there a way to minimize the use of fertilizer and chemicals so that their harmful effect on the environment are reduced? Use of genetically modified crops is a possible solution. Plants, bacteria, fungi and animals whose genes have been altered by manipulation are called genetically modified organisms, GMO. GM plants have been useful in many ways. Genetic modification has: 1. Made crops more tolerant to abiotic stress, cold, drought, salt, heat. 2. Reduce reliance on chemical pesticide, pest resistant crops. 3. Help to reduce the post-harvest losses. 4. Increased efficiency of mineral usage by plant. These prevent early exhaustion of fertility of soil. 5. Enhanced nutritional value of food. Example, golden rice, that is vitamin A enriched rice. In addition to these uses, GM has been used to create tailor-made plan to supply alternative resources to industries in the form of starch, fuel and pharmaceuticals. Some of the application of biotechnology is agriculture that you will study in detail are the production of pest-resistant plant which could decrease the amount of pesticide used. Bt cotton is produced by a bacterium called Bacillus thuringiensis, Bt for short. Bt toxin genes has been cloned from the bacteria and been expressed in plant to provide resistance to insect without the need for insecticides. In effect, create a biopesticide. Example are Bt cotton, Bt corn, rice, tomato, potato and soybeans, etc. Bt cotton, some strains of Bacillus thuringiensis produce protein that kills certain insects such as Lepidopteranus, tobacco budworm, armyworm, Coleopternus, terans, Coleopterals, beetles and Dipterans, flies, mosquito. B. thuringiensis forms protein crystal during a particular phase of their growth. This crystal contains a toxin, insecticidal protein. Why does this toxin not kill the bacillus. Actually, the Bt toxin protein exists in inactivate protoxin, but once an insect ingests the active inactivate toxin is converted into an active form of toxin due to the alkaline pH of the gut, which solubilize the crystals. The activated toxin binds to surface of the midgut epithelial cell and create pores that cause cell swelling and lysis and eventually cause death of the insect. Specific Bt cotton deity toxin genes were isolated from Bacillus thuringiensis and are incorporated into the several crop plants such as cotton. The choice of gene depends upon the crops and the targeted pest, as most Bt toxin are insect group specific. The toxin is coded by a gene Cry1AC named Cry. There are a number of them. For example, the proteins encoded by the gene Cry1AC and Cry2AB control the cotton bollworm. That of Cry1AB control corn borer. Plants, pest-resistant plant. Several nematodes parasites a wide variety of plants and animals, including human beings. A nematode, Meliodegini incognitia, infect the root of the tobacco plant and cause a great reduction in yield. A novel strategy was adopted to prevent this infestation, which was based on the process of RNA interference. RNA take place in all eukaryotic organisms as a method of cellular defense. This method involves silence of a specific mRNA due to a complementary dsRNA molecule that binds to and prevent translation of mRNA silencing. The source of this complementary RNA could be from an infection by viruses having RNA genome or mobile genetic elements transportions that replicate via an RNA intermediate. Using Agrobacterium vector, nematode-specific genes were introduced into the host plant. The introduction of DNA was such that it produced both sense and antisense RNA in the host cells. These two RNAs, being complementary to each other, formed a double-stranded dsRNA that initiated by RNAi and thus silenced the specific mRNA of nematode. The consequence was that parasite could not survive in a transgenic host expressing specific interfering RNA. The transgenic plant therefore got itself protected from the parasite. Biotechnological application in medicine. The recombinant DNA technology processes have made immense impact in the area of healthcare by enabling mass production of safe and more effective therapeutic drugs. Further, the recombinant therapeutic do not induce unwanted immunological response as in common in case of similar product isolated from non-human sources. At present, about 30 recombinant therapeutics have been approved for human use the world over. In India, 12 of these are presently being marketed. Genetically engineered insulin. Magnesium of adult onset diabetic is possible by taking insulin at regular times interval. What would a diabetic patient do if enough human insulin was not available? If you discuss this, you would soon realize that one would have to isolate and use insulin from other animals. Would the insulin isolated from other animal be just as effective as that secreted by human body itself, and would it not elicit an immune response in the human body? Now imagine if bacterium were available that could make human insulin. Suddenly, the whole process becomes so simple. You can easily grow a large quantity of bacteria and make as much insulin as you need. Think about whether insulin can be orally administered to diabetic people or not. Why? Insulin used for diabetic was earlier extracted from pancreas of slaughtered cattle and pigs. Insulin from an animal source though caused some patients to develop allergy or other type of reaction to the foreign proteins. Insulin consists of two short polypeptide chain, chain A and chain B, that are linked together by disulfide bridge. In mammals, including human, insulin was synthesized as a prohormone like a proenzyme. The prohormone also need to be processed before it becomes a fully matured and functional hormone, which contain an extract starch cell, the C peptide. This C peptide is not present in the matured insulin and is removed during maturation into insulin. The main challenge for production of insulin using rDNA techniques was getting insulin assembled into a natural form. In 1983, Eli Lilly, an American company, prepared two DNA sequences corresponding to A and B chain of human insulin and introduced them in plasmid or E. coli to produce insulin chain. Chain A and B were produced separately, extracted and combined by creating the disulfide bonds to form human insulin. Gene therapy. If a person is born with a hereditary disease, can a corrective therapy be taken for such a disease? Gene therapy is an attempt to do this. Gene therapy is a collection of methods that allow correction of a gene defect that has been diagnosed in a child or embryo. Here genes are inserted into personal cells and tissue to treat a disease. Correction of genetic defect involves delivery of a normal gene into the individual or embryo to take over function of and compensate for the non-functional gene. The first clinical gene therapy was given in 1990 to a 4-year-old girl whose adenosine diamidase deficiency. This enzyme is crucial for immune system to function. The disorder is caused due to deletion of the gene for adenosine diamidase. In some children, ADA deficiency can be cured by bone marrow transplantation. In others, it can be treated by enzyme replacement therapy, in which functional ADA is given to the patient by injection. But the problem with both of these approaches that they are not completely curative. As a first step towards gene therapy, lymphocytes from the blood of the patient are grown in a culture outside the body. A functional ADA cDNA using retroviral vector is then introduced into these lymphocytes, which are subsequently returned to the patient. However, as these cells are not immortal, the patient requires periodic infusion of such genetically engineered lymphocytes. However, if the gene isolated from marrow cells producing ADA is introduced into cell at early embryonic stage, it could be permanent cure. Molecular diagnosis. You know that for effective treatment of a disease, early diagnosis and understanding its pathophysiology is very important. Using conventional method of diagnosis, serum and urine analysis, etc., early detection is not possible. Recombinant DNA technology, polymerase chain reaction, PCR and enzyme-linked immunosorbent assays, ELISA, are some of the techniques that serve the purpose of early diagnosis. Presence of a pathogen, bacteria, virus, etc. is normally suspected only when the pathogen has produced disease symptoms. By this time, the concentration of pathogen is already very high in the body. However, very low concentration of bacteria or virus at a time when the symptoms of the disease are not yet visible can be detected by amplification of their nucleic acid by PCR. Can you explain how PCR can detect very low amounts of DNA? PCR is now routinely used to detect HIV in suspected AIDS patients. It is being used to detect mutation in genes in suspected cancer patients too. It is a powerful technique to identify many other genetic disorders. A single-stranded DNA or RNA tagged with a radioactive molecules probe is allowed to hybridize to its complementary DNA in a clone of cell followed by detection using autoradiography. The clone having the muted gene will hence not appear on the photographic clip because the probe will not have complementary with the muted gene. ELISA is based on the principle of antigen-antibody interaction. Infection by pathogen can be detected by presence of antigen, protein, glycoprotein, etc., or by detecting the antibodies synthesized against the pathogen. Transgenic animal that have had their DNA manipulated to process the express and extra foreign gene are known as transgenic animal. Transgenic rat, rabbit, pig, sheep, cow and fish have been produced. Although over 95% of all existing transgenic animals are mice, why are these animals being produced? How can man benefit from such modification? Let us try and explore some of the common reasons. 1. Normal physiology and development. Transgenic animal can be specifically designed to allow the study of how genes are regulated and how they affect the animal normal function of body and its development. Example, study of complex factors involved in growth such as insulin-like growth factor. By introducing genes from other species that alter the formation of this factor and studying the biological effect that result, information is obtained about the biological role of the factor in the body. 2. Study of disease. Many transgenic animals are designed to increase our understanding of how genes contribute to the development of disease. These are specially made to serve as model for human disease so that investigation of new treatments for disease is made possible. Today, transgenic models exist for many human diseases such as cancer, cystic fibrosis, rheumatoid arthritis, and Alzheimer's. 3. Biological product. Medicine required to treat certain human diseases can contain biological products, but such products are often expensive to make. Transgenic animals that produce useful biological products can be created by the introduction of portion of DNA or genes which coded for a particular product, such as human protein alpha-1 antitrypsin used to treat emphysema. Similar attempts are being made for treatment of phenylketonuria, PKU, and cystic fibrosis. In 1997, the first transgenic cow, Rosie, produced human protein-enriched milk 2.4 grams per liter. The milk contains the human alpha-lactobacterium and was nutritionally a more balanced product for human babies than natural cow milk. 4. Vaccine safety. Transgenic mice are being developed for use in testing the safety of vaccine before they are used on humans. Transgenic mice are being used to test the safety of the polio vaccine. If successful and found to be reliable, they could replace the use of monkey to test the safety of batch of the vaccine. 5. Chemical safety testing. This is known as toxicity or safety testing. The procedure is same as that used for testing toxicity of drug. Transgenic animals are made that carry genes which make them more sensitive to toxic substance than non-transgenic animals. They are then exposed to toxic substance and the effect studies. Toxicity testing in such animal will allow us to obtain result in less time. Ethical issue. The manipulation of living organisms by human race cannot go on any further without regulation. Some ethical standards are required on evaluate the morality of the human activity that might help or harm living organisms. Going beyond the morality of such issues, the biological significance of such things is also important. Genetic modification of organisms can have unpredictable results when such organisms are introduced into the ecosystem. Therefore, the Indian government has set up organizations such as GEAC, Genetic Engineering Approved Committee, which will make decisions regarding the validity of gene research and the safety of introducing gene organisms for public service. The modification or usage of living organisms for public service as food and medicine sources, for example, has also created problem for patent granted for the same. There is growing public anger that certain companies are being granted patent for product and technology that make use of genetic material, plant and other biological resources that have long been identified, developed and used by farmer and indigenous people of a specific region or country. Rice is an important food grain, the presence of which goes back thousands of years in Asia agriculture history. There are an estimated 2 lakh variety of rice in India alone. The diversity of rice in India is one of the richest in the world. Basmati rice is distinct from its unique aroma and flavor, and 27 documented varieties of basmati are grown in India. There is reference to basmati in ancient text, folklore and poetry, as it has been grown for centuries. In 1997, an American company got patent right on basmati rice. The US patent and trademark office. This allows the company to sell a new variety of basmati in US and abroad. This new variety of basmati had actually been derived from Indian farmers' variety. Indian farm basmati was crossed and semi-dwarf variety and claimed as an invention of or a novelty. The patent extended to functional equivalent, implying that other people selling basmati rice could be restricted by the patent. Several attempts have also been made to patent use product and processes based on Indian traditional herbal medicine, example turmeric, neem. If we are not vigilant and we do not immediately counter this patent application, other countries and individuals may encash on our rich legacy and we may not be able to do anything about it. Biopiracy is the term used to refer to use of bioresources by multinational companies and other organizations without proper authorization from the countries and people concerned without compensatory payment. Most of the industrialist nations are rich financially but poor in biodiversity and traditional knowledge. In contrast, the developing and the underdeveloped world is rich in biodiversity and traditional knowledge related to bioresources. Traditional knowledge related to bioresources can be exploited to develop modern applications and can also be used to save time, effort and expenditure during their commercialization. There has been growing realization of injustice, inadequate compensation, and benefit sharing between developed and developing countries. Therefore, some nations are developing laws to prevent such unauthorized exploitation of their bioresources and traditional knowledge. The Indian Parliament has recently cleared the second amendment on Indian patent bill that takes such issue into consideration, including patent term, emergency provision, and research and development initiative.